Hi, everyone. Thanks for coming this morning. It's been a wonderful workshop, so thanks so much for organizing it, and thank you so much for inviting me, and thank you to all the speakers this morning. That was such an inspiring session for me, maybe because I really care about behavior. Today, I'll be talking about frontal contributions to flexible decision-making in rodents. So I'll first talk about executive control of behavior. I feel like just mentioning that in the same sentence as rats is maybe offensive. I'll explain that. In rats, this is my old work requested by Timo. I'm going to talk about that. And then I'm also going to talk about ongoing, very preliminary behaviors in my own lab where we use mice to study strategic decisions in a multi-agent environment. Okay. Executive control, what do I mean by that? Well, here I'm simply referring to the ability to flexibly switch sensory motor routing or mappings on the time scale of seconds or less to match the appropriate environmental context. For example, most of the time when you hear a phone ring, the automatic response is to pick it up and answer. But since you're at a talk, you can quickly form the response of silencing the phone when you hear a ring. But as soon as you step out of this room, you can instantly change your response to the same stimulus based on the new context. And this kind of stimulus-response mapping can be flexible and extremely rapid, and it can be studied in the laboratory using tasks such as the Stroop task. I'm sure many of you guys have heard about that. Subjects are instructed to either read the text of each word or name the color of each word. For example, this word stimulus corresponds to orange in the word reading task, but to blue in the color naming task. Subjects can switch between these two tasks on a very fast time scale, and they display interesting behavior asymmetries where, as you might already intuitively have realized, that word reading is easy and quick and what we call prepotent or reflexive, whereas color naming is slower and harder. To understand the underlying neuromechanisms, we set out to develop analogous behavior in the rat. Stroop will be hard. We were very inspired by the primate pro-anti-saccade paradigm in humans and non-human primates. A lot of its work came from Stefan Everling's lab, and we, inspired by that, developed a rapid task-switching behavior where rats also experience two types of trials, pro or anti. The trial type, maybe the timing of this is going to make some people happy. The trial type is indicated by a sound at the beginning of each trial, and then a silent delay will follow, during which rats have to remember the trial type. So there's no sensory stimulus during the delay period. There's no spatial information during the delay period. There's only the rule or memory of the rule. After the delay, a target stimulus will be presented as either the left or the right light. If it's a pro trial, rats have to orient towards the light for reward. If it's anti-trial, rats have to orient away from the light for reward. And the rules on each trial, the stimulus side on each trial, completely interleaved across trials. It was obviously not an easy task for us to train the rats to do, but it was very important that we got this correct in the sense that rats can perform fast rerouting between the pro and anti-task on a trial-by-trial or single-trial basis. We were happy to find that not only can rats do this, they actually displayed very interesting behavior asymmetries between the pro and anti-task that are very similar to what's observed in analogous primate paradigms, such as in Stroop or in the pro-anti-saccade task. So first, pro is faster to learn than anti. You can see pro can be acquired within a single session. Anti took a few days. And really interestingly, only the rats that first learned pro managed to go on and perform pro-anti task switching within session, as if the anti alone first rats, like after they learned this hard behavior, they became inflexible in learning. But we followed up with the first cohort of rats, of course, because they actually learned the task. And also, pro has better accuracy than anti on average across animals. Obviously, there's some variance across the, different individuals. And of course, pro is faster to execute than anti. And all of these, I should again mention, that are consistent with what's being observed in the primates. And I don't have time to go into this, but this is extremely interesting. We actually have a whole paper about this, which is switching out of the anti-task, which is hard, right? Actually leads to a larger cost than switching out of the easier pro task, which actually is counterintuitive, and there's a lot of theories about that. stay tuned. If you're interested, talk to me afterwards. But I don't have time to really go into that. So these suggest to us that these asymmetries, not only can rats do this with a task with face validity to primary paradigms, but they also display these interesting asymmetries, suggesting that maybe as in primates, the pro-orienting in our task in rats is also more reflexive or prepotent, whereas the anti-task might be more cognitively demanding. So to understand the neuromechanism, we tested two brain regions that have been implicated in the pro and anti pathway respectively, the deep layers of the superior colliculus and the medial frontal cortex. The SC received direct sensory input, and the corresponding motor projections, these are the deep layers, are essential for orienting to the contralateral side, motivating the idea that SC is important for the stimulus-driven pro pathway. On the other hand, both the primate and rodent literature hypothesize that the medial frontal cortex encodes the relevant task goal and allows anti-performance. This is important by directly inhibiting the pro pathway via suppression of the SC activity. This very specific hypothesis for how anti can occur, referred to as the inhibition model, is supported by a wealth of evidence. Human PFC lesion study, human neuroimaging studies, monkey single unit recording study, all in the pro-anti-saccade paradigm. So we tested this model by doing pharmacological inactivation of medial frontal cortex and SC using muscimol, which is a GABA agonist that lasts for a few hours. And we predicted, based on these, that inactivating the SC should impair pro performance and facilitate anti, and inactivating medial frontal cortex should impair anti performance. And indeed, unilateral inactivation of the superior colliculus resulted in a motor impairment contralateral to the inactivated side, whereas uni-PFC, I don't have time to show you today, but resulted in a general anti-impairment. So these unilateral inactivation data seemingly are consistent with the inhibition model, but we really have to be careful interpreting unilateral inactivation data because you have to think about hemispheric competition as well as hemispheric compensation between the two hemispheres, right? And when I did bilateral inactivation of the SC deep layers to directly test its causal role in the task, the pro deficit that was seen before after uni activation was actually rescued when the two hemispheres are balanced. But maybe even more surprisingly, we saw a major anti-impairment. And based on past literature, we were expecting the SC and PFC to have the inactivation to have the opposite effects on behavior, but instead we actually found similar results. Arguing against the idea where at least the idea that SC needs to be inhibited by PFC for anti-performance to occur. So I was asked not to show too much neural data, but since Andreas and Sandra showed all these beautiful neural, I actually just now added this slide, so might be a little bit rusty in presenting this. So this is actually recordings. So we did single unit recording, PFC and SC, when rats are performing the task. And what I'm showing you here is each row is a neuron, and the color represents the pro versus anti-selectivity. And this is D prime, but very similar to the AUC RC analysis Andreas presented before. And I'm plotting all of the neurons, and the timing of the color will represent the timing of pro-anti-selectivity for this neuron across the trial duration. You can see this kind of, neurons are selective for an anti at different time points, but definitely this is the delay period, right? And you see there's no sensory stimulus during this period. Animals are fixating, there's no motor plan or motor execution, but we have activity that we have activity first, and then we have activity that represent differentially pro versus anti trial. And these are single neurons that I'm not going to have time to go into. But for the same neurons, we can plot selectivity for the IPSI versus contra choice, right? And again, just to accentuate the fact that the delay period has no spatial information, you also do not see any spatial information in the neurons, and the choice information really starts to emerge after the onset of the target stimulus, the left or the right light. Okay, so there's a lot of mixed activity. We actually also did. Thank you, Andreas, for explaining SVM. We use SVM for population decoding as well. Here I'm plotting decoding accuracy. One means perfect decoding of population activity between pro versus anti trials, and 0.5 is chance fuzz because now we're trying to decode between the two. And you can see PFC has a lot of task-relevant rule-related information, but SC actually, shockingly, has even stronger task information, and maybe even more surprisingly, SC had earlier choice information, pretty much as earlier as you can get compared to PFC, even though PFC also has choice information. So all of these, sorry for being the anti-PFC person in a PFC workshop, all of these meant to argue against the idea that PFC and SC play opposing roles during this cognitively demanding task, but instead, they likely cooperate to implement this cognitively demanding anti-performance. So back in 2013, when we got the Museum results, the inhibition model, again, is not about whether PFC is important for inhibitory control. We still think it is. The inhibition model is more specifically about PFC suppressing motor regions to allow NT to occur. This model was so universally accepted that it seems impossible for us to really challenge it, and it will be easier for us to say, well, it's a species difference. But luckily, at the same time, really important paper from Stefan's lab came out where they cooled DLPFC while recording in the superior colliculus, both on the same hemisphere and on the contralateral hemisphere, that provided direct evidence against the inhibition model. And then they wrote this wonderful review paper to re-examine the literature. And actually, there were conflicting evidence along the way against the inhibition model, but we were so convinced by it that we kind of ignored it as a field. So Stefan and Kevin, in this wonderful review paper, proposed an alternative model where PFC sends task-relevant information to the SC to facilitate competitive inhibition against task-irrelevant actions. And we also conducted optogenetics inactivation, physiology analysis, as well as dynamic modeling to follow up and suggest that indeed, key computations underlying executive control could occur within collicular circuits, probably facilitated by PFC projections. Just a little bit of a meta conclusion here. Looking back, our project was really directly inspired by, constrained by, and facilitated by cross-species analysis of cognitive behavior, and especially when the behaviors are really comparable, right, across species, and that was actually pretty important. And finally, I was actually really impressed with how people in the workshop are open-minded about the differences we have and thinking about going forward together. And it was really important, like we are far from being complete in terms of our knowledge of brain functions, and it's so important for us to keep an open mind about, for example, what the PFC does or what any other brain area does, so that when we are faced with conflicting new evidence, we're not just gonna be like, probably just something we can ignore. Okay, so we're continuing with this very interesting behavior in my own lab. We're in a half-X mouse version of the behavior. So now on a pro trial, there's a sound indicating the rule, and the target stimulus will be presented as a left of the right whisker stimulation. Again, the rule cue and the target stimulus will be completely randomized across trials. And then if it's a pro trial, mice have to lick towards the whisker stem side for reward. As an anti-trial, mice have to lick away from the whisker stem side for reward. And my postdoc is more ambitious than I am, so he wants to make the rule also not just two stimuli, but many different stimuli, and there's a threshold that separates the two categories of the rule. And then he wants to make also the sensory stimulus more variable than just the side. So stay tuned for this part of the story. Okay, so for the next maybe 5 minutes, I'm going to switch gear maybe completely to talk about some preliminary, very ongoing, we don't have neural data for it, but based on the literature, we think these behaviors will be very important. For example, to require PFC is a strategic decision in a multi-agent environment. Yesterday, in Susanna's talk, which was so great, she talked about mentalizing about others, right? And I'm never going to argue that mice have the same cognitive ability as humans, right? That's silly. But do they think about others in adaptive behavior in the wild? Of course they do, right? They have to escape predator. They have to think about mating. They have to engage in group foraging. They have to think about territory. So we are very interested in the lab in this kind of decision-making, which reflects, just as in real life, dynamic interactions between multiple decision makers in the environment, and the outcome of one's action depends on the choice of both self and other. So to maximize reward in this kind of environment, you have to, first of all, remain unpredictable, and also to use others' ongoing or sometimes inferred future actions to flexibly guide your own choice. So how do we study this kind of processes? Well, in the lab, we develop different kinds of paradigms. Half-fixed mice playing rock, paper, scissors with a computer opponent versus games, for example, when two freely moving mice engage in competitive social foraging. Hopefully, I'll have time to talk a little bit about both. Okay, first, mouse against computer. Inspired by work by Dale Lee, Alec Kapova, Alex Kwong, we simplified rock, paper, scissors to a two-choice game called matching pennies. Mice are trying to match, and computer are trying to non-match. But essentially, mice lick left or right, and the computer is trying to predict the mouse's upcoming choice based on the mouse's past choice and reward history. So if the computer managed to predict the mouse's upcoming choice correctly, mouse does not get a reward. Doesn't matter if it's left or right lick. But if the mouse managed to remain unpredictable, mouse gets a reward. So can mice learn to become unpredictable against the computer opponent? Well, not on the first day. They mostly lick one side, realizing that there's no reward, and then switch to the other side. This is a very non-flexible behavior, if you will. And when they do this pattern of behavior leads to pretty low reward rate, right? This is very different from the typical learning task. Basically, as soon as the computer can predict what you're doing, you got no reward no matter what you're doing, right? And also, this pattern of behavior, sorry, I don't have time to explain this, but please ask me later on, leads to very low entropy in behavior, and the computer opponent can very accurately predict exactly what the mouse is going to do, right? But somehow, over the course of 1 to 2 weeks, mice, just simply based on reward feedback, managed to become much more stochastic in their behavior. And you can see from this kind of behavior, the choice pattern is much more stochastic, their entropy is quite high, and the best the computer can do is simply guessing. So what is going on during this 1 to 2 weeks process? What are the mice learning? How do they know that this is the right strategy to do in this environment? We are really trying to follow both the behavior data as well as the neural data during this process, both on the mesoscale as well as on the microcircuit level, and to trying to understand mechanisms for continuous learning and sometimes meta learning, as well as stochastic behavior in healthy and down the road diseased brains in the mouse model. Okay, that's it for this, just because of time. What about mouse versus mouse? So to properly test that, we actually designed a new physical arena, what we call the octagon, where freely moving mice can harvest reward from one of the 8 possible sources, hence octagon. So we have floor-projected visual stimuli to indicate where the active patches are per trial. And the patterns of the patches will cue which patch has higher reward or is a higher value patch. So in this case, again, it's grading. So I'm showing you. So this is a trained mouse. This is a top-down view. You see the floor projection of the visual stimuli. This is 2x speed. And the take-home here is that no matter where the patches are in the environment, no matter where the mouse is, when the patches come on, they can very quickly navigate to the high-value patch. And they can generalize this preference across all patch locations. I was quite actually impressed with how fast and how flexible they are in this environment for this value-based decision task. Okay, so the question is, well, what happens when we put 2 mice in there and let them play a simple game? We really went to a very simple game. The rule of the game is that the faster mouse will get the reward and ends the trial. Okay, so the 2 important things. First of all, there's a speed component here, right? The mice are competing, so there is a risk kind of aspect to it, but also there's always two options in the environment, right? High value or high reward, and a low reward option. Importantly, it's not a zero reward option. It's just a low reward option that when they're by themselves, it's the worst option. But when you have a competitive environment that could be fast, you have to consider low versus none, right? So this is a really interesting situation. Now it becomes a multi-attribute economic choice. We have to think about the behavior of yourself and the other animal in order to optimize or maximize for reward. Okay, so I don't know if this is the best example, but you can at least get an idea about the richness of the behavior. Sometimes it makes sense to go for the low reward. And then, because this behavior, as you can see, is very rich and high-dimensional, so sometimes one animal is not paying attention, and you really should take advantage of that for these non-competitive trials within the social session. But as you can see, it's so important for us to really quantify the behavior and characterize the behavior, but we are lucky because now we have a lot of good tools for that. But most of the trials is kind of like the following trial, where mice are engaged with each other, engaged with the environment, and when they see the patches, they race for the high patch. Hopefully you can see even just from this simple video that mice are not ignoring each other during the social session. They're not very asocial. Competition is at least, I don't know about cooperation, but competition at least is very fundamental in their everyday life. And how do we quantify this and what's the result, right? So one idea is that they ignore each other, social behavior is the same as solo, or they race, so they just simply go to the closer patch, and their preference goes to chance, 50%. So what we saw is actually not that. They're still doing value-based decision, but the probability of choosing the high patch is significantly reduced, on average only like 10% maybe during the social session compared to the solo control sessions one day before and one day after. But the interesting thing here is that it's not a general social effect. It's actually quite specific. What do I mean by that? So I only have time to give you one example of this, but there's many examples, so again, happy to talk about it afterwards. So most of the time, the patches can show up anywhere in the environment, right? I'm plotting trajectory for all of the choices within a session. You can see they go to 8 possible reward. But for visualization, we can rotate the trials so that the high patch is always on top, low patch is always to the right, and then it's much easier for us to think about it and visualize it. And then we can plot this kind of things, right? Where I'm plotting probability, which is color, probability of choosing high, blue means 100% choosing high, at different location of the mouse when the stimulus comes on. And you can see that, on average, they're like 80% preferring high, but in most of the position in the arena, they're 100% of choosing the high patch, right? And they only choose the low patch when they're quite far away from the high patch and pretty close to the low patch. Okay, so this is so low. But we can then ask question like, how is the social effect? Is it the same depending on where the mouse is when the stimulus come on, right? And then I told you before, on average, there's like a 10% decrease of probability choosing high. But you can see in the range, in the color here, with the delta p high, is that the range is quite big, right? Sometimes in some position, there's no effect between social and solo. In some position, there's like a 26% difference between social and solo. And indeed, this is not uniform across the location of the entire arena. And then maybe just to give you an example, when the mouse is really close to the high patch, it doesn't really matter if you have a competitor or not in the environment. It's not going to change what your choice should be. It's not going to change the outcome of your choice. You just go for high, right? So mouse behavior is very similar between social and solo. Versus if you're far away from the high patch, then you really have to consider sometimes it's better to choose the safe choice depending on where you are. And I don't have time to show you this, but mice also care about inter-mice distance as well as the distance between other to the high patch. So I feel like we're really excited that we have this novel behavior where we can start to investigate brain mechanism of how mice can integrate self and other information to flexibly guide decision making in a competitive social environment. So really quickly, the Parenti stuff is from my PhD in Carlos's lab, and it's in collaboration with Jeff, Marino, Chuck, and Alex. And I actually didn't have time to at all talk about my postdoc work, which is on half-X mice motor planning, thinking about cortico-subcortical interaction. And the work that I talked about in my own lab is done by these people. Ollie's my postdoc, who built most of the lab and also is the mouse whisperer that trained them on these really difficult tasks. And Joanna is my graduate student who is leading the Matching Pennies project. And thanks to my funding sources, we just got ERC, which is then rejected by ERC because we're not in Europe, and underwritten by UKRI, which means we have postdoc positions available. So if you're interested in these kind of behavior, please contact me, and thank all of you for your attention.